Hello everyone. So today we are going to discuss about non-cyclic photophosphorylation. So in the last sessions we have discussed that photosynthesis consists of two processes. One is light reaction and another one is dark reaction. So this light reaction is also known as photophosphorylation. And this dark reaction is also known as C3 cycle or Kelvin cycle or carbon fixation reaction. Okay. So light reaction is also known as photophosphorylation. So why it is called photophosphorylation? Because it is induced by light. It is a light driven reaction, that's what is known as photo. And why phosphorylation? Phosphorylation is the process when adenosine diphosphate converts to adenosine triphosphate, ATP. So this process is known as phosphorylation process, and here this phosphorylation process takes place in the presence of light. That's why it is known as photophosphorylation. So in photophosphorylation or light reaction, the light energy is absorbed and converted into the chemical energy, that is ATP, adenosine triphosphate, NADPH, and it releases oxygen molecule. Okay, so this is in light reaction, the light energy is absorbed and converted to the chemical energy, that is ATP and NADPH, and in this process it also releases oxygen molecule. So this ATP and NADPH, which helps in reducing the carbon dioxide into the carbohydrate form in photosynthesis process. Okay. So this photophosphorylation mainly follows two paths. One is non-cyclic, another one is cyclic. Okay, it follows non-cyclic pathway. That's why it is known as non-cyclic photophosphorylation, and also it follows cyclic pathway. That is known as also cyclic photophosphorylation. Okay, so today we will be discussing about this non-cyclic photophosphorylation. Why it is called non-cyclic photophosphorylation? Because it is a process which is light induced and it transfers the electron in a non-cyclic pathway to convert the light energy to ATP and NADPH. That's why it is non-cyclic photophosphorylation. So this non-cyclic photophosphorylation, it starts with the photosystem 2. It starts with the photosystem 2. Okay. Now, there is a special chlorophyll molecule is present in photosystem 2, special chlorophyll A, that is known as P680, because it can absorb the light of wavelength 680 nanometer. Along with it, it also contains some other light harvesting complexes. So this light harvesting complexes and the special pair of chlorophyll A molecule absorbs that light energy. Okay. Along with that, it absorbs an electron. Okay. So from where this electron comes from? It comes from the photolysis of water molecule. According to Robert Hill, we have understood that when in light reaction, oxygen molecule is released. So this oxygen molecule is released not from the carbon dioxide but from the water molecule. So in light reaction, what happens? Water molecule in the presence of light, it reduces and it produces proton, electron and oxygen. Okay. So when one water molecule getting splitted, it produces 2 protons, 2 electrons and 1 oxygen atom. But what happens? This oxygen cannot be stay in its atom form, in atomic form. So it has to be molecule. So to balance this equation, to get one oxygen molecule evaluated, we have to get water molecules split. So when two water molecules splits, we get 4 protons, 4 electrons and 1 oxygen molecule. Okay. So now what happens when this water molecule gets splitted, this electron is absorbed by the photosystem 2. So this photosystem 2, the chlorophyll molecule of P680 of photosystem 2, it absorbs the light. Upon absorption of the electron, it gets excited. Okay, that will denote as P680 star. It gets excited means it stays in its higher energy state. So one molecule cannot be stayed in its higher energy state. So somehow it has to return back to its original state, and for that reason it has to transfer its electron to the primary electron acceptor. So here phosphorylating is the primary electron acceptor. Here phosphorylating acts as the primary electron acceptor. This is the PEA, primary electron acceptor. Okay. So as we know that in photosystem 2 there is a reaction center which is phosphorylating-quinone type of reaction center. So this phosphorylating is a modified chlorophyll which contains hydrogen atom instead of the magnesium atom. Okay. This phosphorylating is a modified chlorophyll and it acts as a primary electron acceptor, modified chlorophyll A, which contains hydrogen atom instead of magnesium atom at the center. Okay, so this phosphorylating acts as a primary electron acceptor. Then this phosphorylating transfers the electron to quinone. Phosphorylating-quinone type of center, so it will transfer to plastoquinone. It is a photosynthetically active quinone present in most of the photosynthetic organisms in the photosystem 2. Okay, so this PQ is denoted for plastoquinone. Now the thing is, this plastoquinone is a photosynthetically active quinone. So there are 2 types of plastoquinone. One is quinone A, primary quinone, another one is secondary quinone. So these 2 types of quinones are bound to the membrane proteins present inside the photosystem 2. Okay, and 2 membrane proteins are present, one is D1, D2. So this primary quinone is bound with membrane protein D2 and secondary quinone is bound to membrane protein D1. When this plastoquinone receives the electron, absorb the electron from phosphorylating, it reduced to produce plastoquinone. So now this plastoquinone is again transfer the electron to a protein complex that is known as cytochrome B6F. Okay. Now first the phosphorylating absorbs, then it transfers it to plastoquinone, then it sends it to the cytochrome B6F. So cytochrome is a protein complex, okay, and it contains heme as the prosthetic group, okay, and this cytochrome are of 3 types depending upon its absorption spectrum, its characteristics and its chemical composition. So there are 3 types of cytochrome. So cytochrome is of 3 types. One is A, B and C type cytochrome. So this in photosystem 2, cytochrome B is present and cytochrome C is present. Cytochrome C is denoted as F. That's why it is known as cytochrome B6F. So this cytochrome B6F absorbs the electron from plastoquinone and transfer it to a small water soluble blue colored protein, that is plastocyanin. Okay, this is PC stands for plastocyanin. It is also a electron transfer protein which is blue colored, water soluble and very small, and it absorbs this electron from cytochrome B6F. So what does this plastocyanin does? It absorbs this electron, and along with the proton, it pumps the electron from the photosystem 2 to photosystem 1. Okay, so it absorbs and pumps it to the photosystem 1. Okay, now also we know that in photosystem 1, the special chlorophyll A molecule, special pair of chlorophyll A molecule is known as P700, because it can absorb 700 nanometer of light. So this P700 has some light harvesting complexes. So what it does, it absorbs some light energy and also absorbs this electron. So when it absorbs this electron, it gets excited just like the photosystem 2. Okay, now it gets excited like photosystem 2. We'll denote that as P700 star. Okay, so when it gets excited and becomes at higher energy state level, to return back to its original state, it again started transferring the electron. So here the primary electron acceptor is a modified chlorophyll, just like the pheophytin. Pheophytin is a modified chlorophyll. So here A0 is a modified chlorophyll. Okay, its P700 transfers its electron to A0, which is a modified chlorophyll, and is the primary electron acceptor for photosystem 1. Now this A0 again sends this electron to A1, which is phylloquinone. Okay, so A1 is phylloquinone. It's a photosynthetically active quinone. Except this phylloquinone and this plastoquinone, there are two different types of quinones or photosynthetically quinones are present in green and purple bacteria. One is ubiquinone, another one is menaquinone. Okay, so now we'll talk about this plastoquinone and phylloquinone. So A1 is also known as phylloquinone and also known as vitamin K1. Okay, and A1 absorbs the electron from the modified chlorophyll, then it sends it to the reaction center of photosystem 1. So what is the reaction center of photosystem 1? It is a iron sulfur type of reaction center. AVS type of reaction center. Iron sulfur type of reaction center, which configuration is 4Fe 4S. Okay, and this AVS type of reaction center again send this to another iron sulfur protein, which is small and water soluble, that is ferredoxin. Okay, this reaction center sends the electron to the ferredoxin. So what does this ferredoxin? It is another iron sulfur protein, which configuration is 2Fe and 2S. 2Fe and 2S type. Okay, and then this ferredoxin again transfers the electron to NADP plus, nicotinamide adenine dinucleotide phosphate. Okay, NADP plus acts as the last electron acceptor in this non-cyclic electron flow. It accepts the electron from ferredoxin and the proton from the photolysis of water. Okay, and the protons from photolysis of water. Then it gets reduced to NADPH. Okay, now we understand that in photophosphorylation we get NADPH and ATP. Okay, here this at cytochrome B65, ADP absorbs one inorganic phosphate and produce ATP, adenosine triphosphate. So at this point we get one ATP, and for this point we get an NADPH. Okay, now what is the location of this non-cyclic photophosphorylations? So if you look to this non-cyclic photophosphorilation, it somehow looks like a sidewise Z alphabet. That's why this non-cyclic photophosphorylation is known as jade skin. Okay, it is also known as jade skin. So now what is the location of this photophosphorylation, non-cyclic photophosphorylation? So first, it includes the photosystems. Okay, non-cyclic photophosphorylation includes the photosystem. So we can say photosystems are present in thylakoid. So this happens in the thylakoid. Okay, but to be very specific, for the photolysis of water molecule, like to produce proton, electron and oxygen molecule, this for the photolysis of water molecule, one complex is required, that is oxygen evolving complex. So this oxygen evolving complex is present in the luminal surface of the thylakoid. Okay, if we consider a double membranous structure of thylakoid, okay, this oxygen evolving complex is present here. Okay, the luminal surface or the inner side of the thylakoid. Okay, this is oxygen evolving complex. That means photolysis of water takes place. So this oxygen evolving complex, photolysis of water takes place. A proton and electron produced, and along with this release of oxygen also takes place. So this oxygen is released in the luminal part of the thylakoid. Now there is another enzyme is required for the production of NADPH from NADP+. That is known as FNR. FNR is a flavoprotein enzyme which catalyzes the reaction of NADP+ to NADPH. This FNR is known as ferredoxin NADP+ reductase, FNR. So this FNR enzyme is mostly found on the outer surface of this thylakoid membrane. Okay, so now we can say that splitting of water and oxygen evolution takes place in the inner thylakoid, and NADPH productions happens in the outer surface of thylakoid. So this is the location. This makes us understand where this photolysis of water takes place and where this NADP+ reduced to produce NADPH. Okay, so this is the location. Now the question is, another question is, when we two water molecule is getting splitted, when two water molecule is getting splitted, along with 4 proton, 4 electron is getting reduced. Okay, 4 electron is getting produced. So this photosystem can absorb only 1 electron at a time. So how it manages this 4 electron? What it does, it acts as a gear wheel mechanism. Okay, it's as a gear wheel type. This oxygen evolving complex, it has 5 states. That is ACE0 to ACE4. Okay, oxygen evolving complex has ACE0 to ACE4. So these ACE0 to ACE4 act as a gear wheel type. Okay, what they does, this ACE0 absorb all the 4 electrons from photolysis of water. Okay, 4 proton, 4 electron and 1 oxygen molecule. So H0 absorbs all these things, 4 proton, 4 electron and oxygen molecule. Then it sends 1 oxygen molecule to the photosystem and 3 electron to the S1. Okay, now this H1 will send 1 electron to the photosystem and send 2 electron to H2. Similarly, H2 will send 1 electron to photosystem and send another 1 to H3. Similarly, it will goes to H4, and at H4 it will release only oxygen, and all these 4 electrons will be evaluated or absorbed further by the photosystem. So this is known as A state mechanism. Okay, this is known as A state mechanism. So today we have discussed that photophosphorylation process. It follows two pathways. One is non-cyclic pathway, that is non-cyclic photophosphorylation, and cyclic photophosphorylation. In non-cyclic photophosphorylation, photolysis of water takes place. It produces proton, electron and oxygen. This electron is absorbed by photosystem 2. It gets excited, then it transfers to pyophytin, then plastoquinone, then cytochrome B65, then plastocyan into photosystem 1. Again, photosystem 1, upon absorption of the electron, it gets excited the chlorophyll molecule. Again, it transfers it to the modified chlorophyll molecule, to phyloquinone, to the reaction center, FES type reaction center, to ferredoxin, and then from ferredoxin, the NADP plus accept that electron and reduce to NADPH. So we got the NADPH, ATP and oxygen evolution from the oxygen evolving complex. So this is for today's discussion. If you have any doubt regarding today's discussion, please comment down below. And don't forget to like, share and subscribe. Thank you so much.